Mendelian disease today, particularly features of Mendelian diseases, and then also the current progress in searching for new Mendelian diseases. Okay? Next. All right, so in my mind, there's always been this fascination amongst geneticists at trying to find the gene for X, right? And it goes all the way back to Gregor Mendel. He didn't call them genes, he called them factors. But what he was really trying to figure out was what explained the different phenotypes he saw in pea plants. And I think he must have been quite gratified when he worked out his ratios and so forth. Obviously, that's a far cry from finding the gene, but it gave him additional insight into how biology is working, how inheritance is working. So it's always been exciting to try to make the connection between the inherited material and the phenotype. Next slide. As we talked last week, variation in our species is extensive. There are about more than 120 million single nucleotide variants have been described in our species, and more keep being added every day as we learn more about the genomes of individuals from other parts of the world. As I told you, each of us has about 4 to 5 million single nucleotide variants versus the current reference. And they occur with a spacing of roughly 1 per every 300 to 500 base pairs. They're more common in the non-coding region and less common in the coding region. And just FYI, it's interesting, this amount of variation in Homo sapiens is actually rather low compared to some of our primate relatives, where the variation, the incidence of single nucleotide variants approaches 1 in 100 instead of 1 in 300. I don't know if that's because we're a relatively young species or what. And then, of course, we have another big category of variation, copy number variants. Each of us has 3 to 7 large copy number variants. And 5 to 10% of us have at least one copy number variant that's over 100 kb. And 1 to 2 of us have a copy number variant greater than 1 base. So a lot of variation. And some of it, or a good bit of it, goes unnoticed, but some of it really perturbs the phenotype. Next slide. And I think you've seen this before, the so-called gradient of phenotypic effect versus variant frequency. And we know that if we look at all variants across the genome and plot their frequency from rare to more common, and then compare that to the extent in which that variant can perturb the phenotype, the phenotypic effect size. So the variants that are able to perturb or that do perturb the phenotype extensively are quite rare in general, while those that have relatively little effect on the phenotype are much more likely to be common. Okay. So next slide, or hit it again. So of course the Mendelian variants with large effect size, strong subjection to selection are rare. Next. While those variants that contribute to complex traits have modest effects and are only weakly susceptible to the effects of selection. Okay. All right. Now, an interesting thing about genetics is, and particularly about Mendelian genetics, is we consider the family as our unit. This is quite different from other parts of medicine, where the family unit is really very little considered. We have to beat on people to get them to take an informed family history. Family history, of course, is the cheapest genetic test that one can lay their hands on. It's sitting right across the table from you. But in genetics, particularly Mendelian genetics, we think of the family as our unit. So we're always wanting to know about brothers, sisters, mothers, fathers, and so forth. And we typically use a 3-generation pedigree when we're starting in clinic to try to figure out a patient's problem. That is, the patient, the parents, and their grandparents. Next. Okay, so what are Mendelian traits? They are those traits in which variation in a single gene is sufficient to produce the phenotype. And as we've already said, most of these variants are rare and have a major effect on the phenotype. And they follow Mendelian patterns of segregation. Autosomal dominant, the heterozygote has a phenotype. Autosomal recessive, both copies, both the maternal and paternal copy of the gene are abnormal in some way. Patients can be true homozygotes, that is to say they have both copies of the gene have the same variant, or patients can be compound heterozygotes, in which both copies are abnormal but the variant's different. So you inherit a different variant from your father and from your mother. And then X-linked, what Kirby was just talking about, males are hemizygotes. We only have one copy of the X and one copy of the genes on the X. Females have two, but they are mosaic. That is to say, they're a mosaic of two cell populations. One population in which the paternal X is the active X, and the other population in which the maternal X is the active X. So they get to see the benefits of both copies, but both copies don't appear in the same cell. So if the gene product is one which is put into the extracellular fluid and taken up by other cells, then these two populations can cross-correct each other. But if the gene product is cell autonomous, then they don't cross-correct, and the female may have a phenotype due to the extent that they have a population with the X-bearing deleterious allele active. Everybody clear on that? Okay, next. And here's the scorecard. I think this is the same as I showed you last week. And you should all know your way around OMIM. If you don't, just go in there and search your favorite phenotype or your favorite gene and try out all of the things that you can click through and read about your favorite gene or phenotype. And what you should know is the raw data for OMIM is the literature. They don't take stuff that they hear about, let's say, at a meeting where it's a presentation, and the paper hasn't been written yet. They only take stuff that's gone through peer review and is in the literature, okay? So they tend to lag behind a little bit because that whole process maybe takes 6 months or a year. And generally, they require that in most cases, they like to see 2 papers confirming the result. So it has that little bit of secondary review to help validate what goes in. OMIM also doesn't keep track of an exhaustive list of all alleles causing a particular phenotype. If you go in there, you'll see a few alleles, and those are usually the first allele reported and alleles of particular interest, let's say a vitamin responsive allele or an allele that is particularly bad or something like that. But you won't find an exhaustive list of all of the alleles in OMIM. And about 250 phenotypes that are coming into OMIM every year, so the number just keeps going up. Okay. Now, we might consider, as a general feature, what is the burden of Mendelian disease? And of course, you all know that although the phenotypes are quite rare, by quite rare I mean anywhere from 1 in 5,000 to 1 in a million, if you aggregate them all, they're relatively common. So most present in the pediatric age range. Why is that? It's because they're, in general, pretty severe perturbations of normal development of physiologic homeostasis. So you see them early in life. Not all of them, because for many that are well known in the pediatric age range, there's a tail. If you look at the spectrum of severity of the phenotype, and there's the mild end of the tail extends all the way into adult life. And they are relatively, those at the mild end of the phenotypic spectrum are relatively underdiagnosed in adult medicine. Can anybody think of any reasons why that might be so?
Multi-factor hits. So like if you only have a mutation in one gene, and that's heterozygous for that, you only need one random mutation as you age in a second.
I'm not talking about anything fancy here, Anthony. I'm talking about straight Mendelian inheritance where the individual has variants in both copies, inherited variants in both copies. They're just alleles that have a more modest effect, so the phenotype doesn't usually become quite as noticeable until the individuals get older. Also, as I've already told you, internists generally don't think about Mendelian disease, and they don't take a careful pedigree, and the pedigree is harder to get because the individuals don't manifest a phenotype till they're older. So they may have a younger brother that is 10 years younger and hasn't even gotten to the age where, so it's not recognized that you have multiple cases in the same subshape. Okay, if you go after it with all those thoughts in mind, you can generally tease it out, but it's not nearly as obvious as in the pediatric age. The key point here, though, is that a large fraction, I don't know how big it is, but a large fraction of Mendelian phenotypes that we've learned the phenotype in its severest form in the pediatric age range has a milder tail in which the individuals have milder alleles, the phenotype is not as bad, and therefore they don't present until later in life. Okay. The incidence overall is thought to be around 0.4% of newborns, roughly 1 in 200 newborns. So it's not that rare. There are, last time I checked, 55,000 births a year in the state of Maryland, so you can do the math. That's a lot of new Mendelian disorders coming into the state of Maryland. And in surveys that people have looked and said, okay, let's take the inpatient population in a typical hospital, pediatric inpatient population, what fraction of those patients are inpatients because they have a diagnosis of a rare Mendelian disease? And the number generally is around 10%. And if you go up to the intensive care unit, it's higher because these phenotypes tend to be higher. So these disorders are rare, but if they make up 10% of the pediatric inpatient population, that's not so, you're going to see a lot of it, okay? Next slide. Okay, now let's consider for a moment the attributes of dominance and recessiveness. Of course, these terms came up by who put these names in the literature? Janae?
I'm not sure. Can I phone a friend?
Sure.
Hello, friends.
Gregor Mendel, right? He said certain of the traits in pea plants were dominant and certain traits were recessive. So these are straight out of Mendel. Imagine doing an experiment about 150 or 200, almost 175 years ago, and coming up with a principle that's still used today, virtually unchanged. Pretty good. Okay, next. So they're generally terms describing the segregation of the phenotype. Mendel came up with these terms. He didn't know anything about genes, as we already have talked about. So he simply said, does this trait appear as if only one copy of a factor was present in the plant? Okay, next. So then if you think that, you say, well, what determines the phenotypic consequences of a variant? Why are some variants tolerated in the heterozygous state with no phenotype, whereas other variants in the heterozygous state cause a problem? Okay, next slide. So part of it has to do with the variant itself. What is, you might call that the molecular pathology. Is it a loss of function variant? Is it a gain of function variant? Is it a dominant negative? And we've talked about those terms before and understand them. If it's a loss of function variant, it can be inherited as a dominant, but often loss of function variants are recessive. Gain of function more likely to be dominant. Dominant negative more likely to be dominant. Next. The second big variable that influences dominance and recessiveness is the nature of the protein. Is the protein which is encoded by the gene part of a multimeric protein, or is it a monomer? And depending on that equation, you can influence the expression of a lot of, if it's a multimeric protein, the mutant variant that's a subunit of that multimeric protein can have its effect sort of magnified because it has to interact with the other subunits. Next slide. And then a big variable feature is the demand of the biological system. In other words, the biological system that the protein product functions. So does the biological system demand 100% activity of that protein? Is it, by contrast, is it able to compensate for 50% reduction, so you know nothing? So then that phenotype is going to be inherited. Obviously, you see no phenotype. So that means that problem will be, that variant will be inherited as recessive because the biological system can compensate for it. Then you can imagine some systems that do just fine with 50% activity of a given component of that system most of the time. But then if the system has put a lot of stress on the system, all of a sudden 50% is not enough. And we'll talk about that in a patient a little bit. Okay? So the variant itself, the nature of the protein product, and the demands of the biological system are all variables that influence dominance or recessiveness. Next slide. Now these are just some definitions. I'm not going to read through them. You'll have them in your slides. But you should know all of this like the back of your hand: phenotype, mutation, de novo mutation, expressivity, the extent to which a genetic variant is expressed, mild to severe, and then penetrance to fraction of individuals with a genotype known to cause the disease who have the signs and symptoms of the disease. We'll talk a lot about both of these in a little bit farther along. Next slide. Same here, syntene, recombination, linkage, association. You can read those. If you have any questions, ask the PAs. Now let's get back to the family as our unit. Next slide. Go back a minute. I just want to show you, take a break from the slides. I apologize to the people on Zoom, but I'll show you how I take a pedigree, just for your, because you should be able to take a pedigree. So just imagine yourself sitting at a desk and there's a patient across the seat from you. Let's say a young couple and an affected child. Okay. So I start with a pencil that works. Okay, so I say, do you have any other children? So they say, okay, I got 2 other kids at home. So here's my program. Okay. Now one thing I like to do is I like to take the first name of each person, because then you can talk your way around the pedigree. You can say, what about little Amy or Sally or whatever. So let's say this is Joe, this is Sally, Susie. And I put their age up here usually, say 4.
Does that help?
Huh?
Yeah, it does.
Yeah. And this arrow, of course, indicates my proband. The proband is what? What's the definition of the proband?
The infected?
The affected individual.
The person who brings the pedigree to medical attention. So there could be other affected individuals, but this is the person that caused them to show up for evaluation. And then I may ask a 3-generation pedigree, so you just write this out, and it turns out this guy's got a brother, and over here, it turns out she has a sister, 2 sisters. And I do the same thing. I get their first name so I can say, what about so-and-so? And I put their ages here. And I ask, are your parents still alive? And they may say, well, my father's dead. I say, what did he die of? Pancreatic cancer. My mother's still living, but she has high blood pressure. Her parents, my father is alive, he has COPD. My mother is alive, but she has type 2 diabetes. So you're just very quickly assembling sort of a structure of the family and what's going on medically in the family. Okay? And then you may ask some features about Joe, when did he develop whatever the problem was that he's here for, and so forth. And depending on what he has, you may ask questions to try to see if Sally or Susie has a similar phenotype, just milder, not bad enough to get them to medical attention. You know, is there any possibility that Sally also has a heart murmur? Oh yeah, well, the doctor did mention that. So you know, they won't give you the exact story when you ask it. You have to ask very specific questions. So you can just put it all together like that, and then you have it. And then when you find something, let's say you find this person has something, whatever, and you know that diagnosis, disease X. But you know that is a rare dominant, and in order for that dominant to be relevant to your patient, it would have to go through this person as well. She would have to be non-penetrant and so forth. So you say, well, that's interesting, but it's probably unrelated, okay? Or, oh, that could be the exact same thing, just depends. So you'll find information, and then you apply the Mendelian sort of segregation test to say that this information, this more remote person in the pedigree, is probably relevant to my proband, or maybe it's not. It's just something different. Okay, everybody cool with it? So that's how you can sort of build this information together.
Do you ever cross-reference this with patient health information? Say like the aunt has diabetes, but wasn't mentioned in the patient.
Yeah, so yes. Typically physicians don't ask medical information on individuals who are not their patients. Right? I'm not supposed to go into Athg and look up this person's medical record. I can ask the parents, because that's sort of general knowledge. And I can say to the parents, I really would like to know the specifics of the diagnosis of this person. This person can give me permission to get that information. Or they can get it from her and say, she said she had this or that, whatever. That's okay. But I can't, the HIPAA rules don't allow me to prevent me. I'm not supposed to go look at someone's chart. It just happens to be in my neighbor down the street. I want to see why they're in the hospital or something. I can't do that.
Say you have a child that was adopted and doesn't have access to that biological information. Is that clinically easily passable now with the sequencing, or is that raise a really big challenge?
Well, I always say the only person that doesn't have a relevant family history is an adopted child, an adopted individual who doesn't know anything about the parents. They're literally more or less in isolation. Everybody else, the information is useful. Sometimes it's possible to work back through things and find out something about the biological parents.
Mm-hmm.
But, it's actually one of the things that's been changed is Ancestry. Ancestry.com. And well, actually, this person contacted me and said they're a long lost half sibling, and interesting. Oh, isn't this funny? They have neurofibromatosis, just like I do. Something.
Yeah.
So there is some information that's available, but if a person is adopted and doesn't know their parents, really doesn't know their parents, then they don't count. It's interesting that one such person was Barton Childs. He was adopted, didn't know his adopted parents. So he spent his whole career doing genetics, but he didn't know his own family.
How sticky does this situation get when you're dealing with a family of complete half-siblings?
I didn't hear the first part of your question.
Like, how complicated can this get
Oh.
when you're looking at just a family of half-siblings? Like,
It can get complicated. You know, I mean, it's not unusual to see sort of the initial draft of a pedigree on a piece of paper where, you know, cross that out, put over here, and stuff like that. You know, where you made a mistake, or you turn out this person was adopted into the family, it's not really in this pedigree, and all that kind of stuff. So, as you work through it, you'll find additional information. But certainly, if you know someone is a half-sib, you'd want that information because you know how they're related to your program, right? Okay? Try this on each other.
you don't want my pedigree. It's messy.
All of our pedigrees are messy. All of our pedigrees are messy. Okay, I'll just leave it. All right, let's get the lights again. Okay. Next slide. Now.
It's too slow.
No, go back. Go back. Go back. Okay, forward. I'm missing a slide here somehow, but in the older days, finding genes responsible for phenotypes was quite a time-consuming business. Okay, so how would you do it? You'd try to find a big family that had your phenotype of interest, or multiple families with your phenotype of interest. Even though it was rare, it might take you months, weeks, years to find that. And then use linkage, let's say, to try to find the region of the genome that was likely to harbor the variant that was responsible. Then you would try to say, well, I think it's on the long arm of chromosome 7, but you wouldn't have had any markers on chromosome 7, or just a few markers on chromosome 7. If you did linkage, you might narrow it down to a region of anywhere from 5 to 25 megabases, so lots of genes in there. You could then sequence every gene by Sanger sequencing. It might take you a year or two to do that. So it's a slow process and relatively low volume. So the big change, really, from my point of view, is this paper. And this is the paper that showed that you could use a sort of genomic approach to disease gene identification. And this is a group from the University of Washington, Seattle. So they asked, they just started to do whole exome sequencing, okay? And they said, I wonder if we could find the gene responsible. Let's just take a patient that has a rare syndrome. This syndrome is called Freeman-Sheldon syndrome. And by the tedious approach, it was already known to have mutations in this gene, MYH3. So they said, okay, let's take a patient with a known mutation and see if we can find it by whole exome sequencing. Okay? Now, they didn't have all the databases that we had now, but they sequenced this patient, Freeman-Sheldon syndrome 24895. And they found 4,500 potentially deleterious variants in the exome. They said which ones were rare. They're not in DB-SNP. Still a lot, 500. They did 8 HapMap exomes. The HapMap population was relatively healthy. They said which ones in my patient were not in 8 HapMap exomes, about 800. They took both of these and combined them. Which one were in neither of these two things? They were down to 360. Which ones were predicted to be damaging? 160. So that's 160 genes that might house the variant that was responsible for this rare syndrome. So what did they do next? They made the assumption that there was no locus heterogeneity, that everybody with this syndrome had a mutation in the same gene. Right? They said, if that case, let me do it with a second patient. So they got another patient with Freeman-Sheldon syndrome. They went through the same thing, although they combined the results with both patients. And by looking at those two patients, they came down to only 10 genes that had deleterious mutations that were shared by both patients. Okay? Well, that was pretty good. Let's do it again, right? So they got another patient, did it again, and they came down to 2 genes. And they got another patient, did it again, they came down to 1 gene. And guess what that gene was? MYH3, the gene that they already knew was the right thing. So that said that I can use this, there's no segregation here, no pedigrees, just accurate diagnosis. Now, if the Freeman-Sheldon syndrome had locus heterogeneity, they would have been screwed, right? Because it had 2 patients with the same clinical phenotype but mutations in different genes. And this whole analysis would have fallen apart. The other thing is it depends on accurate diagnosis. I'm always down on dysmorphologists, but I have to give them credit here. They had it right, and if they didn't have it right, this would not have worked. But the bottom line here is it said that you can, by sequencing broadly across the genome, not knowing anything about location of the genome, just knowing something about the nature of the variants, if you have enough patients and the diagnosis is homogeneous, then you should be able to get there from here. Get to the diagnosis, get to the gene of interest. Okay? So it's a very important paper. It's published in September 2009. And that said, we really don't need big families. Just need patients, accurate phenotyping, and then sequencing. Now, of course, if the mutation was not in the exome, this is not going to work because they were just looking at exomes. Okay? Next slide. So, we'll come back to that paper. Somehow I got my slides slipped out of order here. So that method didn't need family history. But as we were talking about family history before, and I've already mentioned that it's often harder to get in for older patients. I want to show you what I think is a very informative pedigree. So this is a patient we actually saw here. I saw the patient with Hans Bjornson, who you should all know. And the patient was admitted to our medical intensive care unit. And the history was as above. This guy was a 35-year-old male. He had a job at Oracle. He had 3 kids, middle-class family. He had a 10-day history of fever and bad pharyngitis, sore throat. He was treated with antibiotics, but he didn't really respond. His doctor switched antibiotics, trying to find one that would work. And then the doctor decided to try, in addition to antibiotics, giving him steroids. Steroids are, of course, anti-inflammatory, so the doctor, I guess, I wouldn't have done this, but the doctor was thinking, well, maybe steroids will relieve the inflammation in his throat, make him feel better. And anyway, he gave the steroids, and 2 days after starting the steroids, the patient began to develop confusion. He didn't know where he was, he couldn't find things. And so he went to an outside hospital where he was seen in the emergency room. And amongst other things that they measured his plasma ammonia. Okay? It's known that if your ammonia goes up, you become confused. And they found a value of 280 micromolar. Normal is 16 plus or minus 6. Sounds like it's elevated. Okay. And so what did they decide? They said, well, this is out of our league. We're going to send him into Hopkins. And the only other thing I'll mention is he had a mild respiratory alkalosis. Ammonia is a central nervous system respiratory stimulant. It's often the measurements of ammonia are sometimes not very good. But if the patient had that degree of hyperammonemia, I would have expected him to have respiratory alkalosis, and he did. So it sort of confirms the validity of the measure. So everything makes sense at this point. He arrives at our emergency room 6 hours later, at which time he's in the early stages of coma. A CT scan shows that he has cerebral edema, swelling of his brain. And his ammonia, as my colleague Saul Bruce Bruce Lowe used to say, has fallen to 420 micromolar, just making the point that it was really elevated. So a pneumonia of 420 micromolar in an adult is good enough to kill you. And he was on his way. This guy was on his way. So we, of course, what did we geneticists ask when we arrived at the NICU, Johns Hopkins Hospital Intensive Care Unit? What's the family history? They don't damn geneticists. By the way, did you see last night, there's the new CBS show, the leading character in the new CBS sort of doctor mystery show is a clinical geneticist. It was the most boring show I've ever seen. You gotta watch it though, it's just so bizarre. They were sitting there, this is supposed to be exciting. He was explaining autosomal dominant. So we said, what's the pedigree? And there was a medical student, and the medical student, they said, well, the medical student got their pedigree, and they showed us this, okay? So we see 2 kids and this man. Now his family was gathering in the waiting room. So we said, well, that's not very good. Are you sure? Go out there and ask for a larger family history. Next slide. So he comes back and, well, there isn't a much more extensive pedigree. So when this shows that the proband had 2 brothers that died in the delivery room, identical twins, died in the delivery room. Urea cycle disorders don't present because you can take care of your mother can take care of your pneumonia. So it takes usually 24 hours to get to trouble. So we thought that was something else. He had a 14-year-old brother that died. So we said, what did we say? Yeah, exactly. What do you mean he died? Most 14-year-old people don't die. Well, he said he didn't ask.
Obviously. Yeah.
So I said, well, go out there and ask again. So he comes back, next slide, and he says, he drowned. Not to worry, he drowned. So what do we say? We said, well, why did he drown? Right? Go back out. Ask again. Come back in. So it turned out the 14-year-old brother, the deceased brother, was on an outward bound camping trip, and his fellow campers noticed that he had a sort of a respiratory illness, and they noticed that in the 24 hours prior to his death, he became confused. They said, he couldn't find his hiking boots and they were right on the ground, and things like that. And they camped by the side of a lake. And the next morning, when they got up, this guy was floating. Looked like he'd gone off the dock. There was a dock there, and he was floating in the lake, drowned. So we, of course, thought that he was confused. In the middle of the night, he got up and walked off the end of the dock and drowned. And so now this is beginning to look like consistent with what kind of inheritance would you say?
Excellent.
Lawrence is excellent. I like it. Could be autosomal recessive, but it's interesting both affected individuals, if this hypothesis is correct, are males. And what if you thought that, what might you ask next?
About his sister, maybe?
Yes. What about his sister? Or what about his mother? If they both have it, they must have gotten it from their mother. And so heterozygous females for OTC deficiency can be completely normal, or they can actually be severely ill. We just had one that died here a few weeks ago. And very often they spontaneously, ammonia comes from protein, and if you ask them carefully, they unknowingly have a low protein diet. And so sometimes you can get that by asking them what their favorite foods are. So. But meanwhile, our patient was going into coma, so we at that point did pursue it more. But we then thought this was very likely to be excellent ornithine transcarbamylase deficiency, for which, although patients, hemizygous males usually present in the newborn period, there is a spectrum of effect, and some are present late.
And this man had this history of severe infection and steroids, both of which will cause breakdown of endogenous protein, so-called negative nitrogen balance, and breaking down of endogenous protein increases the ammonia that has to be detoxified, and it's less stress on the system. So our hypothesis was exactly that. Next slide. We ultimately went on and tested, these two women were carriers, and then put these guys at risk. And they were sort of wild hares living in Colorado, not Utah, but Colorado. And they were not interested in genetic testing, so I hope they're doing okay. It turned out that he survived. It took him about 2 years to recover from this illness. He had to get on holiness, had to reduce the intracranial pressure. We still followed in the clinic. We got the baby teeth of this person and extracted DNA, and he indeed had OTC deficiency as well as this one. That was all done by Ted Hahn, who was a human genetics register. Next slide. But I want to make the point that sometimes when you get a family history, it doesn't just all fall out. You have to be willing to go back and forth. You do the family history, you get some ideas, you go examine the patient. You say, oh, I should have asked about this, go ask again. So I call this iterative phenotyping, okay? Or iterative family history. So it's not a one and done, it's over and over again as you get different hypotheses and you test them. Next slide. So this is standard pedigree conventions. You can read it on the slide. Next slide. More of the same. Next slide. Okay, so that's an overview of Mendelian disease. Now we're going to talk about identifying new Mendelian disease. Oh, so here's the slide. Human Genome Project, disease gene identification is a slow process. If you recall, if you can imagine this, no reference sequence, no databases of mapped variants, no NOMAD, no NCBI database of SNPs or anything like that. Okay, so you identify informative families, use linkage to try to map the gene to a chromosomal region. You look for chromosomal aberrations that might fall in that region and cause the defect. For example, cystic fibrosis was a chromosomal aberration helped in identifying the cystic fibrosis gene. You could also use somatic cell methods. You could then clone, you could use screen genomic libraries, clone the candidate genes, sequence them, use mutations, loss of function mutations to confirm disease gene identity, and then functional studies. Really slow process compared to that multiple exomes that I showed you. Next slide. So, of course, in this whole process, there's this inconvenient reality that each of us differs from the reference sequence by 4 to 5 million variants. So we're going to find a lot of differences between our probands and the reference. And the game is, how do I filter through those 4 to 5 million single nucleotide variants to find the one that is responsible for this phenotype? Right? Next slide. And sometimes you can combine sequencing and linkage. So we actually did this, by we, I mean primarily Nada Sabrea, who's here on faculty, and our colleague David Goldstein, who at the time was at Columbia. David Goldstein came to give a departmental seminar. Nada Sabrea was a student and had lunch with David Goldstein, you know, like you guys do. And he said he was looking for an unexplained autosomal dominant phenotype that he wanted to do use whole genome sequencing to try to find the disease gene. He said that to the 8 or 10 students who were at lunch with him. So when lunch was over, Nada was walking him to the next speakers, and she said, you know, I just saw a patient in clinic, and that has a phenotype that's not diagnosed. Would you do that one? And he said, sure. Send me the DNA. So here's the pedigree. Here's the proband. Had something called metachondromatosis, which is a defect skeletal, a relatively mild skeletal defect. And it looked like an autosomal dominant, this pedigree. You can see it's a huge pedigree. Okay. So David sequenced this person, and of course found a whole bunch of stuff. So the question was, how could we sort through all that? And he said he was good for doing that one whole genome sequence, but he wasn't good for doing a whole genome sequence on everybody else in his pedigree. So we got the idea of doing a SNP-based linkage study, which was much cheaper. And we did SNP-based linkage. Next slide. We genotyped 12 individuals. Next. And there they all are. Those are the ones we genotyped. So at this time, a whole genome sequence was about $7,000 or $8,000, and a whole genome SNP array was about $400. So it was a lot cheaper. And then we used the SNPs to do a linkage calculation across this family. Next slide. And we found 6 regions that had positive LOD scores. Now, what is the LOD score that we typically say is, that's it? What's the number we typically use? A LOD of 3, right? I mean, it's 1,000 times more likely that the disease gene is in this location than elsewhere in the genome. Well, in this pedigree, we couldn't get to a LOD of 3. But what we could do, we realized, is eliminate all of these other non-positive areas. They were all had LODs of less than 0 or less than that. So we only focused our attention on these 6 relatively small areas. And the second one we looked at, we found, and then we looked at the sequence of this individual, we found a mutation in a gene that explained this phenotype. So, and then we found another patient, another family, and they had a mutation in the same gene. Done. Right? So the sort of not linkage that was going to find the answer, but linkage that was going to eliminate large parts of the genome is what we took advantage of here. Next slide. So there were these different strategies, and currently we identify informative families. We get very good phenotype information with the option for iterative phenotyping. Some consents only allow you to talk to a family once. So we always write in our consents that you can come back to the family. We use genomic approaches, multiple resources for filtering variants to identify high-quality candidate positive variants. And then we use segregation of families, frequency and nomad, what's known in OMIM, what's known in model organisms, for example, the Knockout Mouse Project, KOMP, protein interaction, string DB, expression of GTEx, additional individuals from GeneMatcher, reaching out across the world to find individuals with similar phenotypes and so forth. And then real experiments, i.e. functional testing. So you guys are more or less familiar with it, right? Next slide. Next. So of course now a lot of the sequencing is done in clinical labs. And I like to think of clinical sequencing by the target. So if you go to the clinic and your patient is getting some sort of clinical sequencing, in some instances only a single disease gene is sequenced. This would be the example of breast cancer, let's say, sort of. Now we would sequence more than BRCA1, but it's like sequencing BRCA1 in a patient with breast cancer. Or you could use a panel. You know, like for cardiomyopathy, we know of about 40 genes that can present with adult onset cardiomyopathy. So that's the panel that is used for cardiomyopathy. Or you could do whole exome sequencing, sequence the entire exome, which is how much of the genome? 2%. So that's a lot less sequencing. And most of it is protein coding. So we know the rules for how to interpret variants. We know this variant changes this amino acid, or this variant introduces a nonsense, or so forth and so on. So the interpretation is relatively easy. And then the last possibility is whole genome sequencing. We sequence everything, exons, introns, regulatory sequences. The interpretation is much more challenging. Cost is higher, but you got all the information. Okay? Next slide. So how do we make that decision between whole exome sequencing and whole genome sequencing? So exome is cheaper, easier to interpret. A lot of Mendelian disease is in the exome. It's relatively inexpensive. Interpretation relatively straightforward. Large amounts of reference data. There are some problems, however. Not all Mendelian variation is in the exome. Some of it is in introns, and we'll talk about that later. The capture, I'll show show that in the next slide, is incomplete, so you don't capture all exons. And it has a hard time finding copy number variation, distinguishing one copy from two copies. Next slide. So this is how you make an exome, a library to sequence an exome. Okay? So you sonicate the DNA into small fragments, and then you add that solution of genomic fragments. You add something called RNA base. So you denature the DNA, so the DNA is single-stranded, and you add the RNA probes. And this is a reagent you buy, and it corresponds to RNA molecules corresponding to all the exons in the genome. And typically these are strep avid or magnetic beads. So they're called capture probes. So the capture probes are biotinylated or magnified, and making a selection of library fragments. So you get the library fragment plus the probe, and then you wash away the non-hybridized DNA. And now you've enriched your targeting library by about 50-fold over the genome, and that's what you're going to sequence. And you can also make custom capture probes if you want a particular gene that you really want to saturate or something like that. The main thing is that the capture efficiency, the hybridization of the RNA baits to the DNA fragments, is variable and depends on the sequence context. So some exons, particularly purine rich exons, you don't capture quite as well. So you may not get as many copies of that into your sequencing library. And here's a diagram that shows you that. The main thing is the capture is not, it's a little bit sequence dependent, and therefore you may not get a good representation of the entire exome in your sequencing subject. So that makes a whole genome more attractive. Not only do you get everything, but you don't have to worry about this capture step that is sequence dependent for the exons. Okay, you with me? However, it's more expensive, evaluation is more difficult, a lot more data. The rules for interpreting regulatory variants are still poorly understood, and there's less reference data. It's much better now than when I made this slide like 3 years ago. So it's getting better all the time. Okay, next slide. Now, so then you ask the question, okay, what should I be doing, exomes or genomes? And there are a number of studies in the literature where people have compared, let's say, typically the way the study is done is I sequence exomes on a large number of patients. I found the disease gene in 30% of them. Then I take the 70% that didn't yield me any answers, and I do whole genomes on those and see if I find the answer that way, in which case I find evidence that the whole genome is better than the whole exome. Okay? So the latest study that did that was published about 2 months ago from Berlin, that reference down there. And they looked at 822 families with possible Mendelian disease who had been sequenced with whole exome sequence, families, so they typically it would, they sequenced a program or program plus parents or other people in the family. And this was telomere genome, so the whole genome is in quotes because we know that it's a fraction of the genome that wasn't coded. And they were able to solve, of those 822, they were able to solve 218 using the whole genome. So they were able to solve a third, roughly, of those that were negative to whole exome. So that's great. Now, if they went and looked at those that they solved, 61 or 28%, now they're looking at the 218 that they solved. 61, the variant was such that it would only be detected by whole genome sequence, known as an intron or some other regulatory sequence or something like that. So you couldn't expect exome sequencing to find them. 138 could have been solved by exome, but it was missed by exome. And usually these are regions where the capture was poor. And they can go back. Remember that the interpretation of the exome was made with a read that for 30 reads over that region. And maybe they go back and say, in order to call a variant, you have to see it in 15 reads out of the 30. And you go back and you see, well, actually, 2 reads had this variant, but the capture was uneven. And the variant was there, but I thought it was an artifact and didn't call it. With me? So a lot of this is due to the fact that you don't have that capture step. The other problem is, of course, that the exome part of this study was done a few years ago. So some of this is because the exome found the variant, it was interpreted as neutral, and now with more information we know it's positive. So that's just, you always, when you go back and reanalyze exome data still, you find variants that you didn't pay attention to the first time around, but now you know are important. So there's some of that. So the problem is all these experiments are a little clunky in that way. It's not as clean as I'll take 1,000 people, I'll sequence them the exome, sequence the genome all at the same time, and then I'll see which method is really better. So we're still using some exome, some genome, but more and more whole genome. And I think 5 years from now we'll all be whole genome. Would be my guess. And this graph shows the reasons that they missed governor diagnosis and missed by exome. If you're interested, go and read this paper. I find it a very confusing and poorly written paper. It's okay. Next slide. Now, what about adding long read sequencing? Right? All of what I just told you about was short read sequencing. What if you add long read sequencing? Long read sequencing is good at picking up what?
Structural variation.
Structural variation, right? Because short read sequencing, the reads are about 120, 130. So if the variant is a bigger hole in the genome than that, short read sequence doesn't see it or has difficulty interpreting, stuff like that. So these folks who are from Saudi Arabia took 34 apparently autosomal recessive families that has had a phenotype that was segregating like an autosomal recessive. It was not explained from their whole exome sequencing data. And they did PacBio long read whole genome sequencing on this. And they solved 4 of those 34 on the basis of structural variants. That's what you would expect. They weren't really clear, but they solved some of them because of the ability to reconstruct the haplotype on a long read sequence. In other words, short read sequence, suppose you have 2 variants in the same gene and they're separated by 10 kb. Okay, that's entirely possible, right? Short read sequence, you'll find those 2 variants, but you won't know if those 2 variants are in cis or trans, right? But a long read sequence, because you sequence molecules as long as 10 kb, you'll be able to assign phase. So some of them, I couldn't figure out from this paper exactly how many, but a few of them, the analysis was improved because they could assign cis and trans, and they interpreted single nucleotide variants better. And then they also still found single nucleotide variants that were missed in the exome. That when they found them in the whole genome, they went back and could see them in the exome. But in other words, this suggests that sooner or later, down the road, long read whole genome sequencing is what we're going to be using, but it's not now. Next slide.
Is that a cost?
It's long read sequencing, making the libraries is much more difficult. Right? Making the libraries for exome, or actually for a whole genome, you can use blood on a blood spot. For long reads, you have to make special reagents to keep the DNA, avoid shearing of the DNA. So you want molecules that are 10 kb long or 20 kb long or something like that. So I think one holdback is that the library preparation is more expensive. The second is the sequencing and its interpretation are still, I think, less straightforward than short read whole genome sequencing. But I think those are all issues that are going to resolve. You guys are going to resolve it. Okay, trio sequencing, we've gone over this, I think you know that, but this is the way we find data. If you found a variant in this program, this individual, and you didn't find it in the parents, what would you call that? De novo, right? Is it inherited? Yes. This person inherited
or one of those two parents. It's in the gonads of the parents. So people always say they're not inherited. Well, they are inherited. They're just not constitutional in the parent. Now, suppose I told you that your initial interpretation was wrong. You found it in the proband and you didn't find it in the parents. And suppose I told you it was wrong, your interpretation was wrong. Can you tell me how an error would give this result?
Are you saying that the genotype is wrong?
Yeah, the genotype, the result that you get. You say it's not in the mother, not in the father, is present in the child. It's a de novo. Then I say, no, it's not actually.
So I would think either if it actually is there, it could be a somatic mutation shortly after.
Good idea. That's
Or
possible.
you
In which
have
case
some
it was
sort of-
not inherited.
some
In which
sort of-
case the child was a mosaic.
Yeah. Or if it's in an area of poor sequencing ability,
Right.
it could be a sequencing issue.
Right, absolutely. It could be a false positive called in the child, or it could be a false negative called in one of the parents. So you just have to be a little bit careful about that before you swallow it, hook, line, and sinker. Okay? Carter, did you have a question? Okay.
Non-paternity.
Non-paternity, yes, but when you have these data, you can tell right away who's the father and who's not. 7% non-paternity.
How do you in the clinic tell someone that their dad's not their dad?
How do you think?
How do you say that nicely? I don't know.
Generally, when we get the consent, we say that one of the possibilities from this sequencing is to show that the father's not the father. So we bring it up before the sequencing is done. So if the result comes back after the sequencing is done, it's not a big surprise that we found it. The other thing we often do is the counselor will spend a little bit of time with the woman alone. And just say, you know, when we get this result, if the father's not the father, it will show up. Is that okay? And then they may say, that's no problem, he's the father. Or they may say, oh, he's not the father, he knows, it doesn't make any difference. Or anything in between. Of course, if she says he's not the father, then we say, well, why are we sequencing him? Somebody else. Next. Okay, so unanticipated consequences of DNA sequencing. And these are important to always keep these in mind. We'll find, depending on how the sequencing is done, variants of unknown significance, VUS, is a sequence variant of unknown clinical significance. And we will also find at a certain discovery rate, incidental or so-called secondary findings of medical consequence. In other words, let's say we sequence a baby because they have multiple congenital anomalies, and we find a pathological mutation in BRCA1. Don't explain a baby's multiple congenital anomalies. May be relevant to the baby when the baby gets older, and may be relevant to the family. Okay, so that all is covered in the consent. Here are some possibilities. And typically, secondary findings are found with a frequency of around 2 to 4%. Okay? Next slide. Now, the various sequencing approaches, remember, the sequencing target, vary in this regard. So single gene, examples BRCA1, cost is hundreds to thousands of dollars. Less expensive, it's correct. If you got it, you got it. You don't have to go looking for other stuff. There are fewer variants of unknown significance because those single genes have generally been sequenced a lot, so we know a lot of the variants. And no incidental findings because we're only looking at that one. Panel, cardiomyopathy, for example, 25 proven cardiomyopathy genes, hundreds to $1,000 to $2,000. A broader net, less expensive, less expensive on a gene basis, per gene basis. More variants of unknown significance, no incidental findings because all the genes are known to cause this problem. WES, a clinical WES, when I made this slide, I put 5,800. Well, it's about 4,000 to 5,000 right now. Much broader net, a bargain at the number of genes you're looking at. But you will find many variants of unknown significance, and as I said, on 2 to 3% of the time, you'll find incidental findings. Okay? Whole genome sequence, largely a research tool at this time, but more and more it's becoming a clinical reality. More expensive, it's a broader net still, the broadest, harder to interpret, variants and incidental findings galore. Okay? Not galore for incidental findings, still on the order of maybe 3 to 5%, 4%. Okay, so all of that should be covered in the consenting. And you'll see these articles. My doctor got genome sequencing, told me it might answer the problem, but now he comes and tells me he finds all these variants that he can't interpret. All of that should be discussed beforehand, before the sequencing is done. And the other thing is, of course, what's a variant of unknown significance today, if you go back a year from now, somebody we may have got it, we may have figured out what it is. So these data should be reanalyzed, unlike a lot of other clinical data. And they're still valuable. So we need a lot of genetic counseling. Next slide. Now, there's sort of slides that summarize a couple of studies, and I'm only going to do one of them, then you can just look at the others. It's the same kind of stuff. So this was Baylor College of Medicine, the first 2,000 whole exomes they did in their clinical lab. 88% of the patients were in the pediatric age range. They made a molecular diagnosis in 25%, including 58% of the diagnostic mutations had not been previously reported. They were in known disease genes. The function of the mutations was pretty straightforward, but that was the first time we could see them. Less and less of that now because we just sequenced a lot more. The inheritance pattern of the solved cases are shown there. A key thing is at this time, 2012 to 2014, 30% of the diagnoses involved a disease gene that had just been identified within the last 3 years. So progress in disease gene identification is still going on. It's another reason you have to go back and reanalyze the data. Because 2 years from now, a variant we can't interpret, 2 years from now we might know that that's a real disease gene, disease-causing variant, pathological variant. And then, interestingly, 23 of the patients, 4.6%, had blended phenotypes from 2 Mendelian disorders. In other words, we're always thinking Occam's razor in the clinic, and it may be one of the reasons we're having trouble diagnosing someone is because they have not 1 but 2 Mendelian phenotypes, and we're looking at the blended picture of those 2 Mendelian phenotypes, which doesn't compute clinically. Okay, so lightning is struck twice, basically. Next slide. Next slide. Next slide. So those are just 2 other studies. You can look at it yourself. The take-home message is the same right now. Depending on how the criteria you use to sequence someone, the solve rate is on the order to 30 to 50%. That's pretty good. There's not many medical tests that are anything like that. Plus, you can go back to it and use it over and over again. So finding the disease genes, some immediate consequences, connects genes to phenotypes, connects phenotypes to biological systems, normal and perturbed, unravels locus heterogeneity when you have mutations in 2 different genes can cause the same phenotype. It ends the diagnostic odyssey. There's someone looking for a diagnosis for years and not being able to find it. It's the first step towards informed treatment, and it's a big research stimulus bench to bedside. Okay, everybody got that? Next slide. Okay, this is an example of the diagnostic odyssey, the value of the diagnosis. So this is when this slide was made, he's now 41. But at the time I made this slide, he's 38-year-old male followed by me. He had recurrent episodes of lactic acidosis. That's where lactic acid accumulates. His pH in his blood drops from 7.4 to 7.1 or 6, 7. Pretty sick at that point. He had intellectual disability and cortical atrophy, brain imaging. Cardiomyopathy, his heart wasn't pumping the way it should. And he had autonomic dysfunction. He got dizzy when he stood up. He had trouble with constipation, other evidence for autonomic nervous system. His mother doggedly pursued this diagnosis. She really took him to any doctor who she thought might be able to help him. Huge collection of medical records. She really took great care of him and worked very hard to try to figure out what the problem was. So but she didn't have a lot of money, so we couldn't just do genetic testing willy-nilly. So the first thing we did is a mitochondrial genome because it was cheaper, and many of his symptoms suggested problems with his mitochondria. It was normal. So we said, good news, mitochondria are normal. She said, well, what's the diagnosis? And we still didn't have it. So we had to explain a lot of proteins that function in mitochondria are encoded in the nuclear genome. And we got more money, and we were able to sequence the nuclear genome. Next slide. So he turned out to be homozygous for a nonsense mutation in a gene called FBXL4, which I'd never heard of before, but I looked it up, and it had just been described 2 years earlier as the cause for a mitochondrial DNA depletion syndrome, type 13. These are disorders in which the DNA in the mitochondria get less and less and gradually disappear, and the mitochondria stop working. So it fit pretty well with his and I had to tell her, oh, we got the answer. There's nothing I can do about it, but we have the answer. I mean, I will still treat him as a patient who's having problems with his mitochondria, but I can't correct this defect at the time. She was very happy, I think, genuinely happy to know the diagnosis, to know the answer. Even though it was an answer we couldn't do much about. Now, of course, her optimism was justified because now we're coming into genome editing. Maybe we can do something about it. And there's just been a paper reported within the year that showed that FBXL4 encodes a protein on the mitochondrial outer membrane that is recognized by the system that does mitophagy. And this protein inhibits mitophagy. It's a downregulator for mitophagy. So in the absence of this protein, the hypothesis would be that mitophagy is going on in excess. And that might explain his mitochondrial DNA depletion. And it gives us a target to think about some kind of therapy. Now, most of what he has is not reversible, but there you go. So it's very rewarding, even for the family, even if you can't do anything about it, to give them an answer finally. Okay. Next. What did we learn? Next slide. So some lessons from this sort of community effort to find Mendelian disease genes. Contribution, we found a greater contribution of copy number variants and particularly de novo mutations. The idea of de novo mutations, we knew they occurred, but we didn't really realize what a large fraction of disorders de novo mutations can contribute to. We found that the phenotypes we knew, in other words, the phenotypes we said, here's this disorder, here's the phenotype, were almost always smaller, had less features than the real phenotype. Which now, because we could define the disease gene, then we could go look at people who had mutations in this disease gene, we see they have extra phenotypic features. The multi-Mendels, that's people with 2 Mendelian disorders instead of 1, genetic heterogeneity, locus, allelic and locus, variable phenotypic expression. Learned about various things that contribute to that. We identified some druggable targets based on the Mendelian phenotypes, and we learned about what underlies penetrance, not completely, but in some instances. Why some people have the disease mutation but don't have the phenotype. Okay, next slide. How are we doing for time here?
11.
Who?
11:57.
Okay, we'll do just maybe 5 minutes more, and then I'll pick up at this point the next.
We have one more lecture, so we're good.
Yeah. So I think let's just talk about de novo mutations. Remember I said before this all started that is finding disease genes. We underappreciated the contribution made by de novo mutations. So now we know, and I gave you the reference, and I think you're reading it for this week's paper, that there are about 100 de novo mutations depending on the paternal age
And they occur at random, so 1 to 2%, 1 to 2 of them would be expected to hit the exon. Most are of paternal origin and increase with paternal age. Of apparent de novos, 5 to 10% of them are somatic germline mosaicism and apparent. In other words, normally we think for most of these the mutation occurs during gametogenesis. But in some instances, when you go back and look at the parents, they don't seem to have the phenotype, but if you sequence like their blood DNA, you find maybe 5% of the genomes have this variant. So the parent is a mosaic. Mosaicism includes not only the soma, but the germline, and just happen to pass on the germline mutation. That's a low occurrence, but it does happen. And some de novos confer selective advantage in the germ cells. This is amazing. The one for achondroplasia, which is FGFR3 missense mutation. If you look at the abundance of that variant in men who are shown to have had a child with achondroplasia, you go look in the sperm and maybe it's present in 10%. But then you come back to that individual 5 years later or 10 years later and it's actually present in 20 or 30 or 40%. It's as if spermatogonia with this mutation have some selective advantage in the gonad, and therefore the older the man gets, the greater the abundance of spermatogonia with this mutation. It's implicated in neuropsychiatric disease, congenital heart disease, and others. Anything in which the reproductive fitness of the affected individual is reduced, because that means that these variants have to be replaced each generation. They appear to play a role in maintaining the frequency of disorders with reduced reproductive fitness. And there's a big review, next slide, on de novo mutations, which is worth your read. Here's this table. They compare de novos with inherited variants in terms of where they are, their susceptibility to selection, and so forth and so on. Read that chart and think about it, and you should understand those conclusions. Should be apparent to you. Next slide. And there's this graph that I already showed you, showing a linear relationship about, on average, 2 new de novo mutations per year of paternal age. That's all listed off there. Next slide. Now, let me just say, let's do phenotypic expansion as well. This is good. So here's a little girl who's a patient of ours. There is her pedigree. She had an older sister that died. Next slide. So she's a 2-month-old female with dilated cardiomyopathy. Her heart muscle is distended and the heart doesn't pump. It's a problem with a squeeze, as the cardiologists like to say. Next slide. Family history is positive for a sib who died of dilated cardiomyopathy at 8 months of age. So that's suspicious, right? The sib who died had a panel, a dilated cardiomyopathy panel, which included about 30 disorders associated with dilated cardiomyopathy that were known to cause dilated cardiomyopathy and also known to occur in late childhood or early adult life. Next slide. Our patient underwent, for her dilated cardiomyopathy, underwent a cardiac transplant at 7 months of age. Big deal. Did okay. After the transplant, and when our lab was just developing whole exome sequencing, Kim suggested, why don't we do a whole exome sequence on this patient, and we did. And to our great surprise, the patient was a compound heterozygote for ALMS1, a gene that is responsible for something called Alström syndrome, loss of function mutations. So she had Alström syndrome. Now, what's the phenotypic expansion? Next slide. It turns out that it was not known that Alström syndrome caused early onset dilated cardiomyopathy. It was known to cause dilated cardiomyopathy, but usually in adults. So it wasn't on the panel that was used for the older sib. So it was known, but it wasn't on that panel. So the diagnosis was missed
on the older SID, and we went ahead and told them, we think, the older SID must have had, quotes, virus. That's what doctors use to explain when they don't know anything. Must have been a virus. And lo and behold, the virus came again, right? It turned out to be Ulstrom syndrome. So that is expanding the phenotype. We now know that Ulstrom syndrome can cause very early onset cardiomyopathy. And it's tough to know because Ulstrom syndrome includes renal disease, lung disease, intellectual disability, hearing loss, and so forth. And this child who we did a cardiac transplant so that she could live has all of these other problems. All of these other problems are appearing. And parents might have thought twice about doing a cardiac transplant if we had known the right diagnosis. Okay? So, and then this is multi-mentals. This is cases 1, 2, 3, 4, 5, 6, 7 who have mutations in 2 disease genes instead of 1, and the various diseases that they have and the inheritance patterns. You can read about it in this paper in the journal. So you have to always think, I can't make this diagnosis because it has some features of this disease, it has some features of that disease, and maybe they have both. Right? So that's Occam's razor may sometimes be dull. You guys know what Occam's razor is? So it's Occam's razor is some sort of story or legend or whatever that if you can make the simplest explanation possible. And sometimes a simple explanation, that's a good thing to try, but sometimes it's not right. Next slide. Okay, so we're going to stop here and we'll pick this up. We're about 85% of the way through the